My guest today is Professor Trey Ideker, who is a professor of medicine, bioengineering, and computer science at the University of California, San Diego. He directs the National Resource for Network Biology and the Cancer Cell Map and Psychiatric Cell Map initiatives. Welcome, Trey.
Thanks very much. It's a pleasure to be here.
Yeah, thanks for doing this. So I have a number of your papers, at least I have the titles of them. I haven't read them. But they're all extremely interesting topics. As I mentioned, I have done some work in AI, but away from life sciences, more into economics and business. But these topics are of real interest to me. So I think there is sort of 2 or 3 buckets of papers here, but I want to start with a multiscale map of cell structure fusing protein images and interactions. So this is sort of the hardware of the cell, right? I mean, it seems like a very, very complex machinery once you go in there.
That's right. And so this is everything in biology is referred to often as either structure or function. And so I can see you understand, Gill. This is the hardware, the structure of the cell. That we're trying to get at in a systematic way using a combination of data and machine learning.
And so what do you mean by a multi-scale map? What would be the output of this process?
Well, actually, to me, those are 2 distinct questions, or two different questions. They're linked, of course. What is it meant when we say, what do we mean by multi-scale? And then what is the actual output? How do we represent the model and visualize the model? And so let me just start with what do we mean by multi-scale? Back to your analogy here of the machine. It's a very complex machine when we consider a human cell. Some people think in terms of the complexity of the internal workings of a cell, it's on the same order of complexity as the complexity of a brain, right? You can think really, in fact, there's a wonderful book written by Dennis Bray about a decade ago, which I think the title of the book is Wetware. And the idea is that the behavior of cells is extraordinarily complex and exhibits what might be considered intelligent behavior. Right, and so the difference there between a cell and a brain, of course, is the hardware, and maybe some of the software, but certainly the hardware is different. The hardware of a brain can be decomposed into neurons and banks of neurons that are the decision, maybe the atomic processes of the brain as we like to think about the brain. And then what are the atoms of that processing inside of cells? Well, they're proteins and circuits of proteins that are making those decisions. And that's what are in the contents of our models. Now, it's multi-scale because let us think about what are the physical scales of these machines. So we start at the lower reaches of scale or resolution of what we consider with individual letters in proteins, genes and proteins. And if you think about what is an A, C, G, or T in DNA, and what's the size of this, the answer is, well, about a nanometer. Somewhere in the 10 to the minus 9 meters, 10 to the minus 10 meter scale. Of course, proteins don't act alone. So genes encode for proteins, and they have the 3-dimensional structure. Those 3-dimensional structures don't act alone, but they combine with other 3-dimensional structures to form even bigger structures, and these are typically called protein complexes. But a protein complex can be relatively small, of just maybe 2 or 3 subunits, or it can have hundreds. So the size of a protein complex could be anywhere from, say, 10 nanometers to 100 nanometers or more. Okay, an example of a very big protein complex would be something like the proteasome, which is the garbage collector of the cell. It runs around looking for cell machinery, other cell machines that have lived their life and have are either just older older machines that need to be recycled, or defective machines, misfolded machines, and so on. These are collected by this garbage collection unit known as the proteasome, and the proteasome itself is a machine of dozens of different proteins. And so again, this is a bigger machine. And so you know, when you talked about the cell as sort of this complex set of machines, it's very hierarchical, isn't it? Because you have single proteins as the atoms, if you like, or amino acids as the atoms inside of proteins, but then proteins are the atomic units of protein complexes. But these, of course, are just the subunits, again, in a modular way of bigger and bigger machines. If you continue upwards in scale and you get up to organelles, so collections of protein assemblies forming larger assemblies take place in organelles, and these organelles are maybe of the size, say, 1 micron. So 10 to the minus 6 meters in size. And finally, organelles like the nucleus, the mitochondria, the energy factories of the cell, or the chloroplast if it's a plant, the lysosome. These are all examples of organelles at around plus or minus the micron level. And then finally, those are within cells, and the cell itself is the very highest resolution we consider in this paper you read. We're really trying to model one cell. And how big is a cell? Well, for humans, about 10 microns, so 10 to the minus 5 meter. So multiscale, we're talking 10 to the minus 10 or 10 to the minus 9 meters, which would be a change in a nucleic acid or a amino acid of a protein. You get mutations and nucleotide, genome variation that makes us who we are. These are events at the level of 10 to the minus 9 meters. And then the cell itself is 10 to the minus 5 meters in size. And so we have to then span these 5 orders of magnitude. Okay, so that's the multiscale. Now, I haven't yet really answered the full question, which is how do we represent that? And we represent, but now I can say it quite quickly, because everything I've just described can be represented, at least abstractly, as an ontology of parts in cells. And so what's an ontology? Well, most people represent ontologies by graph structures. Sometimes they're trees, but in our case, it's hierarchical, like a hierarchical tree. But it's actually a so-called DAG, a directed acyclic graph, because you allow for reuse of smaller parts inside of multiple types of larger machines. Just like, when you assemble your piece of furniture that you've just bought from IKEA, you may have 46 screws of the same type, and 12 of them go here, another 12 of them go in this part, and so on. And so when you have that kind of repurposing of smaller machines that can be mixed and matched to make up larger ones, you no longer have a tree structure. It's formally a DAG. And most ontologies respect this type of structure. So there are layers of objects, and the objects of a layer draw from the objects below it in the hierarchy for their.
Sort of like a blueprint of a cell. But are we up to a point that we have some sort of a, software or a combination of hardware and software product that, given a cell, it can basically create that tree structure that you talk about because it's all hierarchically organized? So
okay, that last thing you said took me for a little bit of a loop, because let's first talk about function in general, and then you're talking about a specific kind of function, which is how does self-replication happen, right? But let's just talk about function. Let's do something easier, if it's okay to start.
Sure,
how do we start? Because there's plenty of functions. Self-replication, of course, is at the heart of a lot of conundra in both life sciences and artificial intelligence. But let's start with easier functions. So how does the cell decide to grow or stop dividing in response to stimuli? So you can give a human cell growth factors, and all of a sudden it starts to proliferate like crazy. or you can starve it and or stress the cell, and depending on the kind of stress, it'll stop growing and go quiescent is the term we use. Okay, so how does a cell make this decision? And there's many other decisions of this nature, right? That cells, how does an immune cell, right, decide when to go crazy and attack, start defending your body from a foreign invading cell population, versus when does an immune cell, remain quiet, and these kinds of functions. So generally, the way we functionalize this structure gets into another paper you might have in your stack, which relates to, we use these structures as the architecture for deep neural network systems that we use to learn function. So now imagine that we assign every component in the cell that now is in our structural map, right? Every component we assign a bank of artificial neurons in a neural network. Okay, and that bank of neurons, neurons in neural networks have states, ones and zeros. We typically think about the states of any one neuron as output as binary. Of course, they're not strictly binary, but we'd like to think of a train of ones and zeros that those neurons will take on in any given some input and on the way to computing some output. Okay, so now the sort of special thing that we've done is, instead of building deep neural networks to address some input-output translation problem where the neural network is itself a black box, okay, we structure that neural network explicitly according to the hardware that we've just mapped. Okay, and we can get into this, but now what you have is you've added functional state through the states of those artificial neurons onto every component in your hardware. And then we can talk about how it's, and then having that formulation, now we try to make functional predictions. Nothing as complex as self-replication, though. We're just not there yet. But in terms of other kinds of functional predictions, when does a cell proliferate or not in response to growth cues is the main one our papers have addressed. Yeah.
Yeah, it's so interesting, Trey. So I want to take a bit of a detour. So, I mean, the attractiveness of a neural network, a deep learning network, is that you don't actually prescribe its internal structure nor its initial conditions. But when a human goes in there and does that, wouldn't you bring some initial biases into it? Wouldn't that sort of destroy the efficiency?
So it depends how good your prior information is, I suppose, right? And number one. Number two, it depends how strictly you constrain learning using that information. Now, in the first generation systems we've built, it's strict. You're essentially, I like to think about it in a very draconian way. You're taking your otherwise very flexible deep neural network and taking groups of neurons out and literally nailing them to the cross of some predetermined hierarchy, right? That you say this, God comes in and declares these 7 neurons are the neurons that are representing the ribosome, and these 7 neurons are, the proteasome, and these 7 neurons are one of the subunits, are the regulatory part of the proteasome, and these others will be the core garbage collection part of the proteasome, and they then factor and so on. If you're wrong, you're worried about what if you're wrong. Now, I think an opportunity moving forward is to have systems that can take that information, but only under as advisement, and are not strictly constrained. But let's talk about why it's a good idea for a second here. And first of all, I want to suggest it's not such a crazy idea. convolutional neural networks, which of course are the standard now in image processing and image recognition, are themselves built in a very analogous fashion. So what is the prior knowledge a convolutional neural network uses? Well, it partitions a two-dimensional, it assumes its input is two-dimensional, right? As opposed to a standard general neural network, which does not have any preconceived notions about the architecture of the features, right? Here we assume that the input features have a two-dimensional array pattern. And furthermore, we assume that pixels that neighbor one another share information. And based on these two assumptions about the architecture of the features, we then grid the input into these convolutional boxes, the 7x7 box of pixels that I will feed into my first layer, a neuron, and then of course those get through other layers of convolution combined. But again, respecting the two-dimensional architecture of an image and our preconceived notion about that. You can think about our hardware. We're doing the same, just on the two-dimensional or three, or it's the multi-dimensional architecture of a cell.
Yeah.
Right.
No, it's I mean, I love what you're doing here, Trey. So this is a bit of a regime change in the sense that humans have knowledge. Machines are very efficient in looking at information, large amounts of information, and potentially creating insights from that information. Now, this is the problem on the table, I think, for artificial intelligence, which is how do you marry human knowledge, intuition, with machine power, right? I mean, in some sense, that's what you're doing, right?
That's exactly right. And where does it? So let me just get to where it has really been a big payoff so far. So the payoff is an interpretation, right? Which again gets into, so one is how you marry human knowledge about hardware architecture with just plain learning of input-output functions, right? The other is how do you interpret, very much related to your question, how do you interpret the model? And in healthcare, as compared, contrasting healthcare with some other disciplines, in healthcare, you really have high stakes. So in the machine, it's not like, a deep learning system that's predicting what movie you should watch tonight on Netflix. It could be wrong, but you just wasted 2 hours of your life and maybe it was okay, right? Right, the stakes are low, right? At least, I don't know. I mean, for me, 2 hours, okay, next. But if the output is what drug should I give to this patient who is dying of cancer, and if I make the wrong decision, that's the final decision for this patient. I mean, these are the highest, some of the highest imaginable stakes. And of course, there's gradations between these two extremes. So in those applications, you really need the machine to be held accountable. It's got to be trustworthy. That's number one. And so one way of increasing trust, of course, is just like you would with the human. You have to be able to explain your reasons. You can't, it's simply unacceptable for a deep learning system to say, sorry, kiddo, you've got 6 months to live and I can't tell you why. Just can't do it, right? Whereas if you say, I don't really know why, but I sense that you're gonna really want to watch the movie Devil's Advocate tonight. Well, okay, I'll go with that. Let's give it a try, right? So explanations are somehow at the heart of, they're not the only component of trustworthiness, but they're certainly, they seem to be one of the components of trust, of a trustworthy explanation. But the other reason why they're super required, or at least helpful in the biomedical applications we think a lot about, is because then you can start to, if you as a human understand the machine's reasoning, you now can think together with the machine on possibly even better solutions,
right?
Or use that knowledge in some orthogonal task that the machine wasn't directly addressing. So, for instance, if I can predict which patients will respond to a cancer drug, I'll give an example. palbociclib, Pfizer drug. It's a CDK4/6 inhibitor. Those are the proteins the drug inhibits. It's a first line of therapy for certain types of breast cancer. About roughly 30 to 50% of patients do not respond. Okay, so let's say just roughly half patients respond, half patients don't. I'm coarse graining it, I have to assure you, but nonetheless, could I predict which patients are likely to respond versus not, given information that I've collected about that patient, which would include their genome sequence and any mutations that tumor has? We know that mutations cause cancer in different patterns. So are there certain patterns and mutations that would indicate that this patient will respond? And of course, there's many other types of data of interest for prediction. The full clinical record of the patient, is their family history, all of these things, what's their height, weight, and blood pressure. All of these things would be types of inputs the machine could use as features. And you want to predict, is this drug a good idea? No? Well, what about drug B? What about drug C? What about drug D? This is the kind of intelligence system that we and others are trying to build as the next gen healthcare system, okay?
I mean, the beauty of the predictive problem is that you can validate it, you can test it.
And so exactly. So if I say not only this patient's going to respond or not, but I say because of why? Because they have a mutation in the EGFR growth factor receptor, okay, family or something like that, or I have a mutation in the AKT proliferative pathway, right? I can one, index that against my personal knowledge as a physician or scientist and say, oh, I believe that. Okay, I've seen that before as well. But you can also do experiments. You can now go into a dish, perhaps even of that patient's own cultured tumor cells, right? And now try directly knocking out that, interacting and perturbing that mutated pathway in the dish and see it. And so that's going to be, a nice validation of the prediction.
Yeah, it's also risk management, right? So just like you mentioned before, a runaway machine learning, deep learning algorithm could create potential problems for patients. But my bias coming from the other direction, Trey, is, and I'm coming from engineering and business, and my bias is you should let the machines run because humans often bring noise that don't add much to the problem. In my fields, right? I mean, biology is quite different, especially when the stakes are so high. You need some very stringent risk management protocols. I think that is really what you are talking about, right?
I think that's one of the aspects that I'm talking about. Certainly risk management is one of those aspects, and quality control and the prediction, but the ability to validate, right?
Yeah.
But then but now in terms of then future drug development, if I have the ability to look at what parts of the cell in this machine are the key parts for predicting a patient outcome or a drug response, I can now go develop drugs against those parts. Whereas if the machine can only tell me the drug works or does not, I have no insight of that nature, right?
That's right. Yeah. So looking at the internals of the machine has insights for humans that humans could utilize in variety of ways in the future. And so in some sense, machine learning, deep learning outputs, granted, it could be good to use from a predictive perspective, but more strategically, I think the internals of the machines are more useful to create insights, right? I mean, that is to some extent. That's how you think.
I agree. And so maybe there's sort of two types of machine learning applications that we're defining here, and maybe others have defined this already. So there are some applications where the machine learning system really is just learning some functional behavior. So I imagine, and this of course now is outside of my field, But in economics, you simply are trying to do forecasting of various kinds, right? And there's no, it's unclear at least what the structure there is. Let's come back to that, because then in biological applications, of course, often there's this real structure there that you're modeling. And so what you're really trying to get is a structure and functional model of the world. And right now, the state of machine learning is it's very, very heavy on the function. And the structure somehow is optimized fully to learn that input-output function. But I think we're also seeing, of course, an interesting trend even in the functional aspects away from pure optimization of some, minimization of some loss, right? And everyone's now encouraging us, oh, please, community, we know that if there's some competition and some very smart set of people have to think of a way to just tweak that loss a little bit lower, they're going to do it, right? And does that mean they'll get a paper, but does that mean they're going to have an impact? And so I guess my argument would be along those lines. Let's care, yes, let's look at that strict optimization problem, but let's also respect the other dimensions of this, like what is the structure that's actually, what's the structural architecture and the descriptive architecture of the guts of that machine. Of course, brain science, we forget neuroscience that started neural networks originally was doing this, of course. Every neuron that was in the dish was modeled by an artificial neuron in a neural network. And I think eventually, we're even seeing brain science get back to this, where you're modeling both a structure and a function.
Yeah, I was also thinking, Trey, that the human is still the boss. So if I'm an AI black box, my objective function would be ultimately to convince the human of the optimality of my output or my decision. So there is sort of a human training aspect, to the AI. It's not just historical data. It is, how do you convince your boss that you're actually good?
You're taking this to another level. You're leveling up this discussion. I totally agree. And maybe you're getting into adversarial learning, right? Is almost one way you could approach the structural aspect as well.
Yeah, so it's a mathematical question there. I don't know much about it, Trey. I haven't done anything in that area. But I think what you are doing here, ultimately, you're saying all the stuff is good, but at the end of the day, you have to make the right decisions. Not only that you have to make the right decisions, but you have to be able to explain those decisions.
Correct.
Right? And so black box, we build a lot of black boxes in financial markets, and they work for a while and then they crash and burn. That has been the history of financial markets. And that's because we can explain how the black boxes work. They are regime dependent black boxes. And if regime is stable and sustaining, the black boxes work. But if they don't work.
They don't generalize, right?
They don't generalize. But in biology, you have an additional vector there, which is the hardware is knowable in some sense, right? Maybe that is what brings the equation differently.
Well, and in a sense, as a biologist, you care about both structure and function are at the heart of nature. So as natural philosophers, in the ancient use of that word, nature is both structure and function, and it's inherently interesting because it's nature. From a healthcare point of view, which of course is built on top of nature, but it's maybe you could get away with just getting the right prediction, except all of the problems you've just raised I can assure you happen in healthcare. So I think you mentioned switching markets, for instance, and then the thing breaks. That the equivalent in healthcare is switching races, ethnicity.
Yeah.
these advanced biomarkers that people build using machine learning techniques do not translate. And what's made even worse is the National Institutes of Health historically has funded research on white people who are predominantly white and often predominantly male, right? And so does it translate to other backgrounds? And many times it doesn't, and of course machines just learn the bias of their human predecessors, right?
Yeah, and then there's a historical bias. I know this, Trey. You know about this. So there was an insurance company in New York who deployed an AI agent that predicted African Americans requires less care based on historic data. And it's because African Americans receive less care. Yes.
Exactly.
And so, yeah, I mean, AI has some difficulties, some growing up difficulties, I would say. I mean, we understand these now, so we won't make stupid mistakes in the future. But we need them, right? I mean, there's so much data out there. We are not utilizing them properly. And so anything that can churn through that data, give us insights, is really what AI is. AI is a guidance mechanism.
Yeah.
It's not a decision mechanism, I think.
That's right. And that's where, when you put it that way, that's where the ability to have a conversation about your reasons and explanations for your decisions is key. Otherwise, you can't engage in a meaningful conversation.
So tell me a little bit about, I was at Pfizer for a little while, Trey, on the business side. I'm always interested in this. So going into cancer, where are we now? You mentioned we could say if a drug is going to be effective for a particular person. In this case, you can assign a probability that the drug is going to be effective for that person X.
Correct.
But are we to a point that this can go into sort of drug discovery to have more of a generalized population-wide optimization?
That question is really on point. So I would argue that we're right there. It's a very interesting time in 2022, nearly 2023 now, where the algorithms still face all the issues, of course, and are struggling with all the issues we've just spent the last 45 minutes discussing. But the solutions are emerging, and there's as you and I both appreciate, progress is occurring here that's very rapid and very interesting. So where are we in terms of using these approaches in healthcare? And there are a few instances where AI ML systems are beginning to be used to match patients to drugs, for instance. Let's talk about where they are being used. So they're being used in imaging. Imaging, I think, has benefited the first from AI ML and its revolution, back to those convolutional neural networks. So everything that came out of Google and ImageNet has been ported into CAT scan interpretation, reading CAT scans and MRIs. There, we could also talk for quite some time about the dangers that have been seen from overfit AIs that cannot explain themselves. But nonetheless, every modern CAT scan, every is a strong word, but there's now many systems that incorporate AI ML into the hardware, actually just sitting behind the hardware for diagnosis. In terms of genetics and genomics, we're just starting to see companies and systems come online, and there the real barrier, in addition to everything we've talked about, is data. So what are the data that we're going to use to train these systems? And let's just talk about that for a second. So the workhorse data generation platform for drug discovery is high throughput cell screening in a dish. Okay, you have panels of millions of cell populations, or maybe just a few, but then on those, you can drop millions of compounds and look at the effect. Okay. And so our systems for drug response prediction have been trained initially from these kinds of high throughput screens in human cell lines growing in vitro, that is to say in a dish. And you can think about it from a feature perspective or from a learning perspective. I have about 1,000 cell lines, each of which has been exposed, call those the rows of the matrix. Each cell line has been exposed to about 1,000 compounds, and those are the columns. And then for each of those 1,000 by 1,000 experiments, I have a growth rate, or what's the effect of the compound on the cell line. And for every cell line, I have many features. It's got a complete genome sequence, so I know all the mutations. For the compound, I know the structure of the compound at the atomic level, and so on. That gives you roughly 1 million training examples right there, and I'm just scratching the surface in terms of what's available in the dish.
And you can do it over and over again, right?
And you can do it over and over again, and you can overfit like crazy as well. Okay. So then even before machine learning came along,
right.
People in biomedicine always worry about translation. And what they mean is, and there's a whole subfield called translational medicine, which is really about how do I take results I see in a dish, or maybe in a mouse, and then maybe I see them in a mouse. So then one next step is towards humans is I stop in mice and rats. Can I now see my effect in a medium scale experiment in rodents? And if that looks good, then I go into human trials. Okay, so it's kind of think about it that way. And this process of translation we know is hard long before machine learning came along. And in fact, if anything, machine learning has some real some wins here. You could imagine in people in machine learning thinking about how you translate or transfer a model across these different contexts, right? So anyway, right. So what's the limitation to finish your question? The answer to your question, we have a lot of data in cell lines. We have a little bit of data in rodents, and a microscopic amount of data in humans. Okay, and so the risk of overfitting on either cell lines or mice is huge, right. And so a conversation that goes on in many cancer centers these days and pharma companies is how I can most expeditiously engage these machine learning systems to start, learning and getting a history of successes and failures in human patients. You have to fail to succeed, right? And get many, and then learn, of course, from those data. But of course, now failures mean patient failures. So that's kind of the catch-22 here is how we start to engage AIs when we don't know if they're any good.
Yeah.
Right.
I mean, I don't know if I read it in your work regularization in biological AI is to some extent is human knowledge, isn't it? I mean, human knowledge can work as a regularization filter on these AI models to make them more useful, right?
That's this medium route forward I was referring to earlier. Don't be so strict about it. Think about it as a regularization.
Right. Overfitting, I mean, we're going to have tremendous problems. I think we are probably 20, 30 years away from actually building an AI system in life sciences, but I think things are moving in the right direction, it looks like.
I don't think I'm more optimistic. I think we're 2 to 3 years away from.
Oh wow! Okay.
Such systems really start to have their first successes.
That's awesome. Yeah.
And certainly within 10 years.
Excellent. So I want to finish up, Trey, with something that's completely different. So I see here is a better way to convert dog ears to human ears, scientists say.
That is completely different. Yes.
So what is that?
Very good. So years ago, a decade ago, essentially, I'll actually give you the historical link to our current topic of cancer cancer machine learning. So one way to try to predict cancer outcomes is using the mutations in the genome. There's other so-called omics layers that one can measure, and another very important type of information is called epigenetics. Or so what is epigenetics? Epi simply means on the gene, so epigenome is on the genome. So there are chemical marks that the same exact genetic sequence can have. So if you look from cell to cell in my body, presumably it's all the same genetic sequence, okay? But the marks on the DNA, called methylation marks, can vary. And this is these marks are partly what determine what cells become lungs, what cells become livers, and what cells become brains, and so on. It's the patterning of these marks that instructs or is involved in the instruction of which genes are turned on and off, and then that creates different developmental pathways. So we were trying to use these methylation. So a methyl group is it's a small chemical group that gets attached primarily to the C's in the AGTC sequence. So the cytosines can have this methyl group or not, depending on tissue type, and depending on lots of different states. And so a lot of people, including us 10 years ago, thought we could use these patterns to predict cancer. Okay, maybe if I see this certain pattern arise now in your bloodstream, and now there's a whole burgeoning field called early cancer detection that actually is trying to do just this, among other things. At the time, the signal for cancer was fairly weak, but the reason it was weak is because there was an overriding signal that governed whether your DNA is methylated or not at a given position, and that was your age. And so what started as a cancer machine learning project, just like we were discussing, there was this magical lab meeting when the grad student, Greg Hannum, who was leading the project, said, guys, I'm going to give up. I've been working at this for a year. Maybe there's a cancer, maybe there's a cancer association somewhere with some part of the genome, but I have to test, almost 1 million different sites in the genome, and that's a huge multiple hypothesis testing problem. And some of them are weakly associated with the incidence of cancer, but that's kind of expected by chance. And furthermore, there's just this big overriding bias. And we said, what's the bias? And he says, PC1 is the age of the patient. PC1, he says, I just can't get rid of, I can't regularize out the age of the patient because your entire measurement, all the methylation marks, or many of them at least, are changing as you age. And we said, really? Well, that's interesting. And pivoted the entire project to publish what was called and is still called the epigenetic clock. So the idea that you can look at this pattern of marks on any patient's genome or any individual's genome, and like a hawker at a fairground, you can get, oh, you're 35 plus or minus 3 years, right? So once you can do that, then the question is, let's look at the people off axis who were mispredicted. Oh, well, by your epigenome, the molecular quantitative measurement tells me you're 35, but you're actually 25. You might want to change something about your lifestyle. You're living fast, man. Or other way around. Wow, you told me you're 50 years old, but you look like you're 25. What are you doing right? So there was that whole angle, and now there's a number of companies, right, that you might have followed in the press based on this epigenetic measurement of age. Okay, but you asked about dogs. So then more recently, we realized, as kind of a fun project, I had a grad student, Tina Wong, who came in and said, Trey, all the stuff with these methylation marks, it's all very interesting, but what about dogs? No one knows their age, actually. And the reason not is because 95% or whatever, 90% of dogs in the US are adopted animals, which means you didn't breed them. So it's your best guess as to how old the dog actually is. And as we got into that, so she spent 2 years creating a dog assay for these marks. And then the more we thought about it, we realized even more interesting than predicting a dog's age is recalibrating the aging curve between dogs and humans. So there's this sort of old adage, 1 year in a dog's life is equal to 7 years in a human's life. And the implication is you have like this just exact line. It's a linear relationship with a slope of 1 divided by 7. Yes. And so what we did is we measured 100 dogs and just used the entire series of marks, because the dog genome and the human genome are so similar. We could literally just align. It's not quite this simple, but it's very simple. It's almost as simple, Gill. You basically just align the genome marks. You say, for any given human, what are the nearest 5 dogs, and how old are they? Yeah. And vice versa, for any dog, what are the nearest 5 humans and how old are they? And from that, you get a very interesting curve. And it's not 1 to 7. It's very, very fast in the early stages of a dog's life. Yeah. So a 1-year-old dog is already aligning to more like a 30-year-old, late 20-something human.
Sort of a logarithmic.
Yeah, so we fit a logarithm. I had more than one person write me and say, Trey, sorry, logarithm is not satisfying. What you want is a power law. It should be a power law, because a log doesn't go through 0, 0. It has to start at 0, 0. And so I agree. There's probably another nonlinear function that's better than we fit. But anyway, I can tell you, if you want to get press for your research, publish on dogs, because I've never all this cancer stuff. But I can't tell you the number of interviews and the number. I mean, I'm in People magazine because of this thing. I mean, much less Smithsonian, Guardian. Paper, Times, it's unbelievable.
No, I was just thinking, Trey, unrelated. I know that we have to wrap this up, but it's unrelated. I wondered this age question. People with dogs as pets and people without dogs as pets. Do humans without dogs age faster? Would be a pretty interesting design question, right? I mean, there might be some data.
I think that might be known, and I think the answer might be yes, but don't quote me on that.
They age faster, right? They age faster.
So I mean, certainly it's known. Yeah. Certainly, it's known that single people age faster than people in marriages. This is an age-old one, and again, I think that probably needs to be updated given society is changing. But it sounds to me very similar to that one, right? Essentially, social connection,
companionship adds. Sort of put some constraint on your aging process potentially.
Yeah, maybe the question is, what's better, a dog companion or a human companion?
Yeah, we don't want to go there. I don't think. I think we'll get a lot of messages and stuff. So excellent. Yeah, this has been great, Trey. I mean, I find your research really, really fascinating. I think this is the only direction to go. And I think the technology is improving, data is getting better. I don't know about the mathematics, though. What's your perspective? I mean, we are sort of stuck on mathematics from the 1960s. I don't really see a lot in the last 50 years. What's your perspective on that?
I mean, my perspective is I hope some smart people will come along and correct that as needed. I mean, my philosophy is it's not going to be me. I live at this bridge of biology and computer science, and the idea is to use the latest stuff, if we're lucky, from computer science to be the first to see the connection into leading problems in biomedicine. And then once we do that, the hope is that we can lure others, maybe through a scientific sense podcast, Gill, into to thinking, who are much smarter on either the computer science side or the biology side, right? And you have both phenotypes there who can really engage and really solve the problem.
Yeah, I mean, most of the innovation, as you know, happens at the intersection of disciplines. I mean, you are at the intersection of two distinct disciplines.
Yeah.
And I mean, I don't want to go on a tangent here very quickly. Do you see some implications for education? I mean, we put kids into this silos. They go become an engineer or a doctor or a lawyer or something. That's all fine, but in the future, I think it is the intersection that matters, right?
I agree, and I don't know if I can weigh in because I see both sides of this one. I mean, are you really proposing that we would abolish siloed departments like I mean, actually, computer science already is a relatively new intersected discipline. It came out of electrical engineering, and so now we have computer science. I have a colleague, Pavel Pevsner, who I think rightly argues that all of biology is basically computer science.
Yes. It's
right?
a computational problem, yeah.
It's essentially it's the life side of computation. Does this mean we should merge these departments entirely, or should we, it's unclear, given that you have to have departmental boundaries, it's unclear what the best way of drawing those lines is, or if one should simply abolish them altogether.
Yeah, I'll be voting for the latter one, but then, I haven't gotten anywhere with that, so.
But then we're just walking around in some embedded latent space of types of strengths and weaknesses we all have, and you're still going to have clusters and affinity groups. Anyway, there's lots of great commentary out there on that already, and I look forward to seeing what people come up with.
Excellent, Trey. This has been great. Thanks so much for spending time with me, and have a great Thanksgiving.
Yeah, you too, Gill. Thanks for the opportunity.
Thank you.